Hi guys, so I've got a fantastic video for you today. I'm going to take you through the full human immune system from start to finish, going through exactly what you need to know for AQA A-level biology. We'll talk about phagocytosis, antigens, antibodies, T cells, B cells, ELISA tests, monoclonal antibodies, you name it, it's in today's videos. Now it's quite a long video today because there's loads to get through and I'm going to go through all the detail you need for AQA A-level biology. So grab yourself a cup of tea, a biscuit, a coffee, pens, stationery, whatever it is you need, and let's get to work.
AQA A-level biology, the human immune system. So let's start with defense mechanisms. Well, the human body has a number of defenses against infectious disease, and these include defense mechanisms like the skin, mucus, cilia, tears, scabs, stomach acid, and the flow of urine. Now, these are all categorized as physical defenses. Defense mechanisms can be non-specific, such as the physical barriers we just talked about, or phagocytosis. There's a diagram of phagocytosis in the bottom right here, and it's basically where a white blood cell engulfs and digests a pathogen. Now, there's specific and non-specific immunity. Well, non-specific immunity involves the physical barriers, which we've spoken about, but specific immunity includes the cell-mediated and humoral immune responses, which we're going to cover in detail soon. So some definitions for you to add to your notes. First of all, lymphocytes, they're a type of white blood cell. You'll learn about T and B lymphocytes in AQA A-level biology. Pathogen is a disease-causing microorganism, and the immune recognition is when a lymphocyte with the correct cell surface protein binds to a pathogen. Now this would be representing a pathogen. It gets taken up via something like phagocytosis into a white blood cell such as a macrophage, and then that antigen gets presented on the surface. So here APC means antigen presenting cell, and then from that we have this coordinated immune response where helper T cells can go on to stimulate B cells and macrophages and T cells and all the stuff we need to fight off an infection. So there is our phagocyte. So phagocytosis, let's go through it in detail. Well, phagocytosis is the process in which a large white blood cell called a phagocyte moves towards, engulfs and digests a pathogen using enzymes. Some products from this process will be displayed on the cell surface of the phagocyte, making it an antigen-presenting cell because it presents the antigen on its cell surface. Antigen-presenting cells help to coordinate the immune response by working with cells, such as T helper cells, to deal with infections. So these are the steps. Write these down in order. So number 1, binding. The phagocyte moves towards the pathogen following a trail of chemoattractants. It will bind to molecules such as proteins on the pathogen using receptors and engulf it, and we can see it here at the top left of this diagram. The bacteria is being bound to by the white blood cell. So step 2, the phagosome. The pathogen is now engulfed and inside a special vesicle known as the phagosome, and we can see that here. So the phagosome is a type of vesicle found in phagocytes, which are large white blood cells that carry out phagocytosis. So number 3, we form the phagolysosome. Well, if you smash the word phagosome and lysosome together, you get the word phagolysosome. So a lysosome is a vesicle containing digestive enzymes, such as lysozyme, and that will fuse, and fuse is a very important term, which is why I've put it in bold there for you, with the phagosome. That will form the phagolysosome. So to summarize, we say lysosome plus phagosome equals phagolysosome. So we see that here we've got our phagolysosome full of digestive enzymes. Step 4, digestion. The hydrolytic enzymes inside the phagolysosome digest and break down the pathogen. Now this is going to allow us to break down and stop the pathogen being harmful or causing any toxins or disorders that may affect us. Number 5, exocytosis. Finally, the digested products are released from the phagocyte, and this is known as exocytosis. Exo meaning outside. And we can see that here just above me. Antigens next of all. An antigen is a molecule such as a protein, but not always a protein. which stimulates an immune response. This molecule could be a protein on the cell surface of a bacterium, for example, which will trigger a sequence of events in the human body that detects it as foreign or non-self. Immunity is the ability of organisms to fend off infection by destroying pathogens, but it begins with the recognition of an antigen. So lymphocytes, next of all, they're a type of white blood cell involved in the specific immune response. Specific meaning we're talking about the tertiary structure of proteins like antibodies and receptors binding to specific antigens. Now, the specific immune response involves the immune system detecting an antigen and dealing with it in a coordinated manner using a range of different types of white blood cells. The bone marrow produces lymphocytes, and these lymphocytes are differentiated further into T or B lymphocytes, depending on where they mature. So I've got a tip for you here, guys. If they mature in the bone marrow, they're classified as B lymphocytes. So B for bone marrow, B for B lymphocytes. If they mature in the thymus gland, they'll be classified as T lymphocytes. T for thymus, T for T lymphocyte. But remember, that's only where they mature. They're all made in the bone marrow, and then they either mature in the thymus if they're T lymphocytes, or the bone marrow if they're B lymphocytes. So B lymphocytes are involved in humoral immunity, and that's immunity involving the body's humours, such as the blood plasma or lymph. Humoral immunity will include the use of proteins known as antibodies, and antibodies have a 3D protein structure and will be specific and complementary to one type of antigen. T lymphocytes are associated with cell-mediated immunity, and this is how the body deals with diseases such as viral infection or cancer. So some definitions next. An antigen is a molecule that triggers an immune response. T lymphocytes mature in the thymus gland and are involved in cell-mediated immunity. B lymphocytes mature in the bone marrow and are involved in humoral immunity. Cell-mediated immunity. Lymphocytes in cell-mediated immunity will deal directly with infected and cancerous cells, as they'll display differing proteins on their cell surface membranes. This form of immunity also explains why organs can be rejected, as they will recognise cell surface molecules as foreign, triggering an immune response. This is an example of cell-mediated immunity potentially causing a life-threatening issue. In short, cell-mediated immunity operates by recognising the antigens on a cell as non-self and triggering an immune response. Now, antigen-presenting cells are cells that display foreign antigens on their plasma membrane. T lymphocytes differ from B lymphocytes in that they exclusively deal with antigens presented on the surface of body cells and not antibodies in the fluids or humours of the body. This is where biologists derive the name cell-mediated, because it directly goes from cell to cell, as we can see in the diagram here. Now, the cell-mediated immune response starts with pathogens invading the body or being taken in with phagocytosis. The phagocytes will then place antigens from the pathogen on their cell surface membrane. Number 3, receptors on a specific helper T cell fit exactly onto these antigens. 4, the attachment activates the T cell to divide rapidly by mitosis and form a clone of genetically identical cells. 5, the cloned T cells will then develop into memory cells, stimulate phagocytosis, stimulate plasma B cells, and activate cytotoxic T cells. So you need to know the 4 key things that T cells will do. Summary: there are many types of T cell. Each one is specific to a specific antigen. The response of T cells to infection follows a path of antigen-presenting cell formation, T cells binding, clonal selection, and the cloned helper T cells then going on to form memory cells. This will stimulate phagocytosis, antibody secretion, and the activation of cytotoxic T cells. So, for AQA A-level biology, you need to know about cytotoxic T cells. Well, these cells are a type of T lymphocyte, hence the name cytotoxic T cells, and that means they mature in the thymus, and they're involved in cell-mediated immunity. They work by releasing perforin, and I always tell my students, perforin perforates. Think about like a perforated letter that has little holes in it. Well, perforin is a protein that kills infected body cells by making holes in the cell surface membrane. Next, The holes in the cell membranes mean that the bacteria or whatever's been attacked by them, like a cancer cell, becomes freely permeable, so it can't regulate substances entering and leaving the cell. And then after this, cell death will result because there's holes in the wall. So in this diagram here, we can see like an infected cell. It could be infected by a virus. Along comes the T helper cell, and it releases porphyrin. That forms pores in the cell membrane of the infected cell, and then the infected cell will die. So the cell will die as a result of the pores in the cell surface membrane. This shows how crucial cell membranes are to the survival of cells. And this action is effective against viruses because viruses replicate inside host cells. So if you kill the cell, you prevent the spread of the virus. So let's do a progress check now. Now I'm going to do this at the end of every topic. And what I'll do is I'll give you a question, then go through the answers. So you might want to have a go at answering these questions in your notes, and I'll give you the answers straight after each question. So question 1, what is phagocytosis? The answer is an example of the non-specific immune response, where pathogens are engulfed and digested by phagocytes. Question 2, where do B lymphocytes mature? The answer is in the bone marrow. Question 3, what is the name given to cells that display antigens on their cell surface membrane? The answer is an antigen-presenting cell. Question 4, what is the role of Thelper T cells? The answer is they develop into memory cells, activate cytotoxic T cells, stimulate phagocytosis, and stimulate B cells and plasma B cells to divide and secrete their antibody. So humoral, immunity, and B lymphocytes. Well, once phagocytosis has occurred and antigen-presenting cells have been formed, this will lead to T cells stimulating B cells. This is where the humoral stage of the immune response takes place. And humoral immunity, as mentioned previously, involves the body's fluids, B cells, and antibodies. Humoral immunity is a stage of immunity that involves antibodies, which are proteins with a specific 3D structure that are soluble in both the tissue fluid and the blood, and you need to know all about antibodies for AQA A-level biology. Now, antibodies will be secreted by plasma B cells in response to a specific antigen. Each antibody is specific and complementary to one type of antigen. And the way in which antibodies are produced is very much a trial and error process. So to facilitate this, there are millions of different types of B cell, each producing an antibody with a different structure. So it stands to reason that eventually one of these antibodies will be the correct structure to bind to the antigen in question. So once we've found out which antigen and antibody we need, clonal selection occurs to produce large numbers of genetically identical cells that all secrete the same type of antibody complementary to the same type of antigen. So in this diagram, we can see clonal selection at work, and we can see 3 plasma B cells, all with different shaped antibodies. So we've got one here that's kind of a blunt-ended antibody, we've got a square-ended one here, and a triangular antibody there. Now the antigen has a triangular shape, so it's only going to bind and form antibody-antigen complexes with the plasma B cell at the bottom. So once the immune system's identified the correct plasma B cell, that specific type of plasma cell will undergo clonal selection and replicate, forming many plasma B cells with the correct antibody. So just to summarise, once the antigen is bound to the corresponding antibody on a B cell, it will then enter the cell via endocytosis and become presented on its cell surface membrane. T helper cells will then stimulate this specific B cell to divide via mitosis, producing clones that all produce the required antibody. As each clone produces the same specific type of antibody, these types of antibodies are referred to as monoclonal. And it's a bit of a silly example, but I always give my students, think about a monobrow, you'd have one eyebrow. Or monotone is one tone. So monoclonal is a single clone of one specific antibody with one specific tertiary structure. Remember that for your AQA A-level exams. So plasma cells. These are cells that secrete antibodies, usually into blood plasma, which is where the name comes from. And these cells survive for only a few days and produce antibodies at a rapid rate during this period. Each plasma cell can produce about 2,000 antibodies per second of its lifespan, and these antibodies lead to the destruction of the antigen. That's incredible. Now, the primary immune response, this is the production of antibodies and memory cells, because there's no memory cells in advance of this. The memory cells are then responsible for the secondary immune response, and here a much more rapid production of antibodies will occur in response to the same antigen the next time it's encountered by the immune system. And memory cells live far longer than plasma cells, sometimes for decades. Now, this explains why you can have immunity for long periods of time. So here's a graph showing the primary and secondary immune response, and we can see we've got concentration of antibody on the y-axis and time on the x-axis. Now, you've got your initial exposure at the beginning, and we have a slow and steady increase in antibody concentration, which takes a fairly long amount of time, or a relatively long amount of time, and it doesn't reach a massively high peak concentration. Now, after the infection, the initial exposure, the antibody concentration falls as time increases, and then we have a secondary exposure, but notice how it never returns to zero, and that's because of the presence of memory cells. Now, during the secondary immune response, the memory cells will divide rapidly, and the antibody concentration, which we can see, will increase dramatically at a much faster or steeper rate, and it will reach a higher peak concentration of antibodies. So, number one, initial exposure. This will be the first time that the antibody has encountered the antigen. Phagocytosis, the formation of antigen-presenting cells, T helper cells stimulating plasma B cells, and the formation of memory cells will be taking place for the first time. Number two, this is the primary immune response, so the rise in the concentration of antibodies in the blood is at a slower rate, and it does not peak at as high a concentration as in the secondary immune response. Number three, secondary exposure. Now, as we mentioned, this is the second time the body encounters the same antigen. Now, notice how the antibody concentration never reached zero, which we said before, and this is due to the presence of those all-important memory cells. Finally, the secondary immune response. So the rise in antibody concentration is more rapid and peaks at a higher concentration. This is due to the presence of memory cells, and this will ensure that the infection is destroyed before it can cause any harm. Now, in your A-level exam, you may have to describe this graph, you may have to explain this graph, you may have to describe and explain it. Now, remember, in A-level biology, the first kind of half of your marks comes from knowing your content, knowing the knowledge, and the second half of your marks comes from analysing and evaluating that knowledge and being able to apply it to exam questions. So, B cells in humoral immunity. Well, number one, the surface antigens of an invading pathogen are taken up by a B cell. Next, the B cell presents the antigens on its cell surface membrane. Then, helper T cells bind to the displayed antigens and activate the B cell. After that, the B cell will now divide by mitosis, forming cloned plasma cells. Next of all, the plasma cells formed will produce specific and complementary antibodies for the pathogen's antigens. Number 6, the antibodies produced will bind to the antigens on the pathogen, destroying them. And 7, memory cells will be formed, facilitating the secondary immune response if the same antigen or pathogen is encountered again. So let's do another progress check. Again, I'll give you a question, pause the video, answer it, and then I'll give you the answer. So number 1, what is humoral immunity? The answer is immunity involving body fluids and antibodies with the action of B cells. Number 2, what is clonal selection? The answer is producing one type of B cell that produces one type of antibody that is specific to one type of antigen. Number 3, what are monoclonal antibodies? The answer is one specific type of antibody that's been produced by a cloned plasma cell. Number 4, what is the role of plasma cells? The answer is that they're cells that secrete antibodies into the blood plasma. Number 5, what is the role of memory cells? The answer, they're responsible for the secondary immune response. Question 6, what is mitosis? The answer is cell division, which leads to the production of daughter cells with identical DNA. Finally, question 7, what is the secondary immune response? Well, the answer is that it's faster than the primary immune response due to the presence of memory cells. So antibodies, next of all, you need to be able to draw these, label them, understand their structure in detail. So an antibody is a protein with a specific structure that is only complementary to a single type of antigen. They're made up of heavy and light chains which have variable regions. Anything other than the variable region, though, is known as the constant region. And we can see that in the diagram here. So in pink, we've got the variable portion at the top, and we've got the constant portion in blue at the bottom. Now, the variable regions will vary dependent on the sequence of amino acids within them, which will therefore dictate the bonds between variable R groups. Check out my video on proteins for more information on that. Now, this will then give the variable region a specific secondary and tertiary 3D structure. So in total, antibodies are made up of 4 polypeptide chains. Once the antibody binds to the complementary antigen, it will form an antigen-antibody complex. Make sure you know all about that because it's a buzzword you need to know for AQA, A-level biology. So, if you've made it this far, guys, don't forget to give the video a thumbs up. We've still got plenty more content to go in the video, but it's been a big one. I know it's been a big one today. So, like, comment, subscribe, and let me know if you like this format of video and I'll make more for you. So, binding of the antibody to the antigen will lead to a number of effects, including agglutination. And this is where bacterial cells become clumped together. So I always tell my students to think about gluing together because agglutination, it's got the word glue within it. So what will happen there is this will act as markers that stimulate phagocytosis. Antibodies can also neutralize toxins. Now, toxins are damaging proteins secreted by pathogens, and if they're neutralized by antibodies, that'll stop them from causing harm. Some definitions for you to add to your notes. Well, the antigen-antibody complex, which remember that's key, that's when the variable region of an antibody has bound to a specific antigen. Agglutination is when antibodies stick pathogens together, and you can see a diagram of that happening here with our Y-shaped antibodies and our little antigens there. And that's going to make phagocytosis a much faster process because you've clumped together all the pathogens. The variable region, now each different antibody has a specific variable region, and it's only complementary, as we've mentioned, to one type of antigen. So antibody structure next of all. Draw this diagram, guys. You need to know it. You need to know about the disulfide bonds, the heavy chain, the light chain, the variable region, all of that good stuff. So the light chain is the shorter type of polypeptide chain found in an antibody. And you can see that on the edges here and here. So an antibody has 2 light chains. The constant region is the part of the protein that will not change, meaning it is common across all types of antibody. The receptor binding site is where the receptor on the lymphocyte will bind, and the antigen binding site is where the antibody will bind to the antigen, forming an antigen-antibody complex. The variable region will differ depending on the type of antibody and depending on the R groups of the amino acids that make it up. The heavy chain, this is the longer one in the middle, and there's 2 of them in each antibody, and it's made up of, amino acids, which is why it's called a polypeptide. Monoclonal antibodies, next of all, are a single type of cloned antibody, and they can be isolated for use in science and medicine. Now, you can use them for COVID tests, which we've certainly got a lot of experience about recently, pregnancy tests, and also the diagnosis of diseases such as HIV, influenza, hepatitis, and chlamydia. Now, magic bullets using monoclonal antibodies to target the medication. This is used in cancer research. So cancer cells are particularly difficult to treat because they're human cells. So they're going to have very similar proteins to a lot of the cells in the human body. And the things that will kill them will kill other cells in the body. So basically what we need to do is target drugs so they only affect the cancer cells. Well, monoclonal antibody therapy is a process where the first step is to produce monoclonal antibodies, and that's so that they're specific to antigens on the cell surface membrane of a patient's cancer cells. Now, after this, these antibodies are administered to a patient and they bind to the receptors on cancerous cells. Now, finally, the antibodies will bind to the antigens on the cancer cells, meaning that it will block the chemical signals that can lead to increased cell division and tumour formation. Now, when treating breast cancer, Herceptin is an example of a monoclonal antibody used. This has big advantages over treatments such as chemotherapy and radiotherapy because the monoclonal antibodies lead to fewer side effects because it's not damaging the healthy tissues as much. So indirect monoclonal antibody therapy is where a cytotoxic or radioactive drug is associated with a monoclonal antibody specific to cancerous cells. So this will target the drug towards the cancer cells, leading to them being killed. And again, this will lead to a reduction in side effects which would otherwise be caused by the drugs. Now, home pregnancy tests use monoclonal antibodies, and you can see it on the right-hand side here, a little pregnancy test. Now, the placenta produces a hormone called HCG, human chorionic gonadotropin hormone. Don't worry, you don't have to know that. Now, this is released in the expectant mother's urine. Now, the monoclonal antibodies will be linked to a coloured compound and complementary to HCG. The HCG antibody colour complex will then move along the test strip until it binds to another antibody which is in a fixed position, and this will indicate a positive result with a coloured line. And we've all got experience of that recently with COVID tests, and we all know that, the coloured molecules move upwards and then they go to the C, which is the control, which means you don't have COVID. But if the coloured line sticks to the bit where it says T, that will indicate you do have COVID because it will have bound to the monoclonal antibodies there. So, if the mother, in the case of pregnancy tests, is not pregnant, no HCG will be found in the urine, and therefore no HCG antibody colour complexes will be formed, giving a negative test result. So producing monoclonal antibodies next of all. Now this can be quite controversial because it does involve animal testing. So it's important to be aware of ethical implications. So on one hand, monoclonal antibodies have been used to treat very serious diseases such as cancer. But on the other hand, we have to use organisms like mice in order to develop them. In addition to this, there are also safety implications to the human volunteers that are involved in drug trials. So how do we form them then? Well, step one is a mouse is injected with an antigen for which the scientists are trying to produce an antibody against. The B cells in the mouse will then start producing antibodies, and they will be complementary to the antigen they've been injected with. So we'll extract those antibodies. Now, number 3, the B cells will then be mixed with myeloma cells, which are a type of cancer cell, and that's because myeloma cells divide rapidly. Number 4, the B cells and myeloma cells are then fused together, and that forms a hybrid oma. Oma because of the myeloma, hybrid because it's a hybrid between the B cells and the myeloma cells. So as I've said, cancer cells are used because they divide readily outside of the body. Now, number 6, the hybridoma cells are separated using light microscopy, and each separate cell is cultured to form clones of itself. And finally, each culture is made up of a single type of hybridoma cells that produce one type of monoclonal antibody, and they'll be then tested to see whether it's the desired monoclonal antibody we want for, say, our pregnancy test or our HIV test or our cancer treatment, whatever we want to use that monoclonal antibody for. So vaccination next of all then. Well, vaccinations work by introducing an antigen directly into the bloodstream via an injection or through oral administration. A vaccination contains a dead or attenuated form of a pathogen. Now, the vaccination will contain one or more types of antigen from a particular pathogen, depending on factors like antigenic variability. After the vaccination has been completed, the individual will produce memory cells which remain in the blood long term and allow for a secondary immune response upon the next exposure to the same antigen. Now, as mentioned previously, this will lead to a more rapid increase in antibody concentration, leading to the pathogen being destroyed, often before the individual shows any symptoms. Herd immunity next of all. So once a large enough percentage of a population has been vaccinated, this will actually give protection to the unvaccinated individuals, and this phenomenon is known as herd immunity. And with enough people in a population immune to a disease, there'll be less people who can pass the pathogen on, basically. So antigenic variability next of all. Well, if a pathogen's got a high rate of mutations, such as influenza or HIV, this is going to lead to antigenic variability. And this is where we have rapid mutations, leading to different antigens on the cell surface, so the antibodies produced previously will no longer be effective. So again, an example of this is found with the influenza virus. So some ethical considerations next of all. Well, there's no doubt that vaccinations have led to millions of lives being saved, and the eradication of deadly diseases like smallpox. This, however, does not mean that there are no factors to consider. So some key considerations you need to be aware of include animal testing, side effects, drug trials, religious views, personal views, and a number of others. So ultimately, it comes down to weighing up the benefits and risks and using scientific data to form an informed opinion. Passive immunity. Well, this is the introduction of antibodies directly into an individual. No memory cells are going to be formed, so the immunity is immediate, but it's not long-lasting. Active immunity. This is when individuals produce their own antibodies in response to an antigen. So natural active immunity results from just being infected with a disease, whereas artificial active immunity results from vaccinations containing dead or weak pathogens. So our next progress check is on antibodies and vaccinations. So question 1, which part of an antibody changes? The answer is the variable region. Question 2, which biological molecule makes up antibodies? The answer is proteins. Question 3, what is agglutination? The answer is when antibodies clump together pathogens. Question 4, what are the uses of monoclonal antibodies? The answer is pregnancy tests, treating cancer, and diagnosing diseases such as HIV. What is passive immunity? The answer, directly introducing antibodies into an individual. Number 6, which type of immunity do vaccinations lead to? The answer is artificial active immunity if you're given the pathogen, but if you're just given the antibody, then it's artificial passive immunity. Question 7, what is herd immunity? The answer is when the majority of a population has been vaccinated and become immune to a disease. Question 8, what is the name given to pathogens which frequently undergo mutations leading to new antigens? The answer, antigenic variability. So HIV, well, what is HIV? HIV stands for human immunodeficiency virus, and it's the virus that can lead to the development of AIDS, which is the acquired immune deficiency syndrome. It works by inserting viral RNA into a cell along with an enzyme called reverse transcriptase. Now, reverse transcriptase, A's because it's an enzyme, reverse because it does the opposite, transcript because it does the opposite of transcription. So basically, it does the reverse of transcription. So it's an enzyme that converts RNA to DNA. And it does that within the target cell. And the target cell in this case is T helper cells. Now, this DNA can then form new HIV molecules and lead to its spreading. So someone who is infected with HIV is said to be HIV positive. However, the development of AIDS only occurs once enough damage to T helper cells has occurred to put them in an immunocompromised position. Now, fortunately, it's no longer the death sentence it used to be. With the modern development of antiviral medication, fewer patients have seriously reduced life expectancies than ever before. Now, due to the lack of healthy T helper cells, though, individuals suffering from AIDS are very prone to infections and cancers, and it's these secondary diseases that typically cause the death of the patient rather than the HIV virus itself. Now, HIV is made up of a lipid membrane, attachment proteins, reverse transcriptase, and a capsid. And you need to know about HIV structure and HIV replication, and that's exactly what I'm going to help you with next. So the structure of HIV, first of all, well, we've got our lipid membrane around the outside. We've got our attachment proteins that are used to bind to the T cells. We've got our reverse transcriptase within the capsid, which is the enzyme used to convert HIV RNA to HIV DNA. And then we've also got the RNA within the virus as well. So HIV replication next of all. Now, this is going to cover a lot of information, so feel free to pause the video and make notes. So step 1, HIV enters the patient's bloodstream and it is transmitted via blood. Number 2, it then binds to a CD4 receptor on a T helper cell. Although it can bind to other cell types, the T helper cells seem to be the most common. So we can see that with number 1, we've got our HIV here, and we can see its attachment protein binding to the CD4 protein on our T helper cell. Now next, the capsid fuses with the T helper cell's membrane, and RNA along with reverse transcriptase enters. Now we can see that at the top right corner here, we can see the capsid entering the host cell and the RNA and reverse transcriptase inside the host cell now. So number 4, Reverse transcriptase, because remember it's an enzyme, so it'll catalyze the conversion of RNA to DNA. And we can see that here. We've got our RNA, reverse transcriptase, forming viral DNA there. Now 5, the HIV DNA enters the nucleus and is inserted into the host cell's DNA. So we can see that there. We can see this yellowy orange piece of viral DNA within the host cell's DNA there. Now number 6, the mRNA is then created, which is used to produce HIV molecules, and we can see them in red there. So that DNA has been transcribed into HIV RNA, and that's then moving through the cell out of the nucleus. So number 7, the HIV molecules then break away from the cell membrane, with part of its cell membrane forming their phospholipid envelopes. So you can actually see the virus using the host cell's membrane to form its lipid envelope. I just think that's so cheeky. They're almost like cellular pirates, if you like, where they commandeer a ship. They take over a host cell and make the host cell produce viral proteins and viral DNA for it. So quite a cheeky mode of operation there, really. So next of all then, ELISA testing. Now this is one of our top tools for fighting HIV because it allows us to identify who's HIV positive and who's HIV negative. So ELISA stands for enzyme-linked immunosorbent assay. And look at this diagram, guys. I mean, beautiful, absolute beautiful diagram. Now, I made it in paint. I mean, no expense spared for you guys. We work hard to get you the grades you need. So enzyme-linked immunosorbent assay. Well, it makes use of monoclonal antibodies and can be used in the detection of HIV. So step 1 is to fix HIV antigens to a plate, such as a glass slide. Number 2 is to add a sample of the patient's blood to the plate, and if they're HIV positive, their blood will contain antibodies specific to the HIV antigen and bind. So I'm just going to take my pen here and just shade in here, just to indicate we've added the patient's blood. So we then wash the plate, and that's going to remove unbound antibodies. So that's that first antibody we can see there, the first Y-shaped antibody. So we've got our HIV antigens at the bottom there, and then we've got the patient's blood containing antibodies if they're HIV positive. So we wash the plate to get rid of them, any of the ones that are unbound. Then step 4, we add a second antibody. And that's an antibody that's complementary to the first HIV antibody. But this second type of antibody has got an enzyme link to it, which I've represented in purple or pink there. Now, if the patient is HIV positive, there will already be HIV antibodies bound to the HIV antigens on the plate. So just to recap, the triangles are antigens, the Y-shaped molecules are antibodies, and then we've got a second Y-shaped molecule, which is an antibody with an enzyme linked to it. And that's where the enzyme-linked part of ELISA comes in. So step 5, we wash the plate again to avoid any false positives. And finally, we add a substrate that will change colour if the enzyme is present. So for example, if this enzyme was lactase, we could add a substrate that would go blue if the lactase enzyme was present. And remember, the enzyme will not be present if it's been washed away. So it will only be present in a HIV positive patient's blood. Okay, so let's do a progress check next of all to see how you're getting on with this. So question 1, what does HIV stand for? The answer is human immunodeficiency virus. Number 2, what does AIDS stand for? The answer is acquired immunodeficiency syndrome. Number 3, what is the name of the enzyme that HIV introduces into a host cell called? The answer is reverse transcriptase. Number 4, what does HIV bind to? The answer is the CD4 protein on T helper cells. Number 5, what does ELISA stand for? The answer is enzyme-linked immunosorbent assay. Number 6, why is the plate washed in between steps in ELISA testing? The answer is to avoid false positives. Okay guys, it was a big video today. I really enjoyed making it for you. If you like the longer format videos that do like entire topics, let me know in the comments. If you prefer them short and sweet and put into a playlist for you for your revision, let me know about that one as well. But in any case, I hope you got a lot out of the video. And if you did, don't forget to like, comment and subscribe. And I will see you in the next one.